Hello everyone, Gauri here. I hope you all are doing well. In today's session, we will be exploring plant hormones. According to a standard definition, plant hormones are small organic compounds synthesized by specific plant cells or tissues which are active in low concentration and promote or inhibit growth and developmental process. The concept of plant hormones originates from a classical experiment on phototropism, the beginning of plants towards light, carried out by Charles Darwin and his son Francis in 1880. The Darwins were able to demonstrate that when oats seedlings were exposed to a lateral light source, a transported signal originating from plant shoot apex promoted differential cell elongation in the lower parts of the seedling that resulted in bending toward the light source. This signal was subsequently shown to be IAA, the first known plant hormone. Based on function or chemical structure, there are five major groups of plant hormones, of which four will be discussed here. These groups are auxins, gibberellins, cytokinins, abscisic acid, and ethylene. Auxin, the first hormone, was discovered by Fritz Wendt as a growth promoting chemical in the tip of oat coleoptiles. Various naturally occurring auxins are known, namely IAA, that is indole 3 acetic acid, IBA, that is indole 3 butyric acid, and PAA, that is phenyl acetic acid. There are more than 200 auxin analogues with different chemical structures known to have the common auxin activity. Major sites for IAA synthesis are the shoot apical meristem, young leaves, and developing fruits and seeds. The physiological effect of auxin includes stimulation of cell elongation or expansion in stems and coleoptiles. The differentiation of relative amounts of xylem and phloem are regulated by auxin concentration. Auxins also support in rooting, apical dominance, tropic response, and fruit set. Fruit set is defined as the transition of a dormant ovary to a rapidly growing young fruit. The second groups of plant hormones are gibberellins. Gibberellins are a group of tetracyclic diterpenoid compounds made up of four isoprene units. Gibberellins were discovered by Kurosawa in Japan in the course of his studies on fungal diseases of rice. There are currently 136 gibberellins identified from plants, fungi, and bacteria. The gibberellins are named GA1 till GAN in order of discovery. The physiological effects of gibberellins includes internodal elongation in genetically dwarfed plant species. A role of gibberellins in flower induction in reproductively competent plants has been established in long day and biennial species. Gibberellins also promote seeds germination by activating vegetative growth of the embryo and mobilizing stored food reserves of the endosperm. Applications of gibberellins can cause fruit set. For example, stimulation of fruit set by gibberellin has been observed in apple. The third groups of plant hormone are cytokinins. Cytokinins are adenine derivatives that have diverse effects on important physiological functions in plants. The first cytokinin was isolated from herring fish sperm in 1955 by Miller and his associates. This compound was named kinetin because of its ability to promote cytokinesis. The first naturally occurring cytokinin was isolated from corn by Miller and Latham independently and it was called zeatin. There are two types of active cytokinins, zeatin and isopentenyl adenine. The physiological effects of cytokinin include the formation, maintenance, and growth of the shoot apical meristem. Cytokinin and auxin stimulate morphogenesis in the callus. Direct application of cytokinins to axillary buds inhibits apical dominance and stimulates the growth of lateral buds in many species. Not just that, chloroplast maturation and senescence are also assisted by cytokinins. Now let's talk about the fourth plant hormone, that is abscisic acid. ABA is a stress hormone and was first identified and chemically characterized by Frederick T. Endicott and his co-worker when studying compound responsible for abscission of cotton fruits. In contrast to auxins, gibberellins and cytokinins, which are represented by various active derivatives, ABA is a single compound. ABA functions as decrease in the size of stomatal aperture and also inhibits stomatal opening. ABA has different effects on the root and shoot growth, and the effects are strongly dependent in the water status of the plant. ABA is required for the development of desiccation tolerance in the developing embryo, the synthesis of storage proteins, and also in the acquisition of dormancy. That is it for today. I hope you enjoyed watching the video. Thank you.